my pleasure to introduce Andrew Thurber. Andrew is a professor at Oregon State University, and some of the work that he's going to be talking about today that was in Antarctic was supported by his recent SF postdoctoral fellowship in Thurber research. He's also one of the leads for the International Network for Scientific Investigation of Ecosystems, INVIE. He got his PhD at Scripps, working with Lisa Levin on role of archaea and food webs, and then he was also, of course, in Los Angeles when he was graduate student, where he and his thesis research here worked on microbial food sources for sponges in the Antarctic. So I think we got Andrew thanking for the rain that he brought down to as well this morning. And I'll let him move forward with his talk.
Great. Well, it's wonderful to be back. I think the last time I was here, there was a different podium, and it was my thesis event, so it's always kind of nice to return back home and how things change, and there's no longer a control team here. But today I'm going to talk to you about bacterial-based food webs. This is something that I actually started working on as my thesis here and has developed into a variety of different avenues since then. And really, what I'd like to look at the role of bacteria in food webs is because it might be a potential food source in the deep sea, potentially very important to the globe all around us. In this figure, which is a map, this color is the deep sea. You can see it's a large percentage of the planet. It's about 64%. And if you want to look at it by scale, here we have area in kilometers squared. We have all of the different habitats you find in deep seas on logarithmic axes. And you can find out they're very large. Since I'm from Oregon, or land of rain, you can see Oregon smaller than that, North America, and even all land Denmark are much smaller than the deep sea. So it's an area where we really need to understand what goes on there and also what it does for us as a society. So as a classic, all deep sea biologists like to put this in their talk. It was in the New Yorker. I don't know why I don't care about the bottom of the ocean, but I don't. And there's a couple of reasons why you should care about the bottom of the ocean, and one of those is its role in the global carbon cycle. And we all know the Keeling curve. We put a lot of carbon dioxide into the atmosphere, and because of the flux into the deep ocean, the ocean processes themselves have mitigated our impact on climate through a variety of processes. I also like to connect this to the Martin curve, which is essentially the flux of a particular organic carbon matter throughout the ocean. You can see it asymptote relatively shallow within the ocean, but eventually there is an important part that's deposited on the seafloor that's about 0.4 to 2 gigatons carbon per year. And what I study at the benthic environment is essentially trying to figure out the processes that control whether that carbon is remineralized, re-released into the overlying water column, which means it's going to come back to the surface on about a 1,000-year time scale or less, or whether it's buried in the seafloor, not broken down by either metazoan, i.e. animals, or microbial processes, in which case it's essentially geologic record and out of sight and out of mind, most likely beyond the scale of human growth planet. So that's one reason why you should care, and also one reason why I like to study the ecology of deep sea habitats. Now, the deep sea is vast, it's also changing. We used, as part of the exercise, we essentially ran 32 of the best carbon or climate models on the planet. They were the most recent IPCC report, and we use it to predict essentially the different stressors, not at the surface ocean where most of us think, but on the benthic environment. So each one of these figures, and the colors go from 0 to 1, there is a value here, but really the important point is the magnitude of stress. And this depicts the impact of changing temperature using a reduced CO2 emission projection. This isn't the flat line projection that many of us think is completely useless because we've already surpassed it every time we rerun a model, as well as the business as usual, which is the most likely scenario. And you can see that temperature is going to shift in the ocean, even at the seafloor. Oxygen stress is going to shift if there's going to be reduced oxygen, mostly for the world over. pH stress is going to be in certain areas where we are not so surprised about. But importantly, food, productivity, the amount of carbon that's deposited on the seafloor, as predicted by these models, is likely to decrease over time. But unlike these, which are mostly unidirectional, productivity in food will shift slightly in different areas. However, if you look at how that might actually impact the different fauna classes, alter the ecology of the deep sea, you can see with these predictions, which I'll say are predictions and emphasize that, essentially these are hypotheses to test, but essentially demonstrate that with our current state of knowledge, the deep sea will likely be impacted over the next 100 years. All of the main faunal size classes of metazoa are predicted to decrease by the year 2100, which is not that far in the future. So the ecology is shifting. How is that going to play out? and the essentially relative balance of carbon getting back into the water column, which is that buried into the seafloor. The other thing you need to know about the deep sea is it's episodic, cold, and food limited. So this is one of the better illustrations of this that I've seen. This is at 500 meters depth off the Antarctic Peninsula. You can see there's seasonal pictures of the seafloor, and then all of a sudden there's a large deposition of hydrothermal food sources, if you're a ecologist like I am, so you think of it, onto the seafloor. So it's not a gentle rain that constantly falls to the seafloor. That occurs, but that's not the dominant export production. It's really seasonal, big pulses that have to sustain benthic communities, both microbial and metazoan, for a long time. I would like to say they came up with a brand new idea, but I actually think some of the old ideas just need to be revisited. This is one of my favorite figures by Lindemann in 1942, and what it does is essentially crosses all size class and integrates an ecosystem into one. I like it because you have ooze, and then arrows essentially connecting bacteria to browsers, what we would now call phasers, benthic predators, swimming predators, predator plankton, zooplanktons, and phytoplankton. In other words, what this presents is that there's an ecosystem where bacterial communities are completely linked to the ecosystem around them, including the food web of those phasers. So, even older, very first issue of Nature, Sir Wyatt Thompson, said there's little or no light in the depths of the ocean, naturally there are no tanks, but the bottom of the sea is a massive animal life. And what do these animals feed? That was in 1870. And my answer to that is, well, it might be microbes. And if it is microbes, that may actually impact the carbon balance, as well as our understanding of the mechanisms that lead to ecosystem functions on the largest habitat on the planet. However, the deep sea is difficult to study, mostly because it's hard to do manipulative work to truly understand how mechanisms occur at the deep sea. And so today I'm going to be talking about a shallow water analog, which in this case is as far south as you can go on the planet without using one of Stacy's fancy ROVs, and that's the Thurber station. Overall, I want to study essentially how does the intersection of microbial and animal ecology dictate the functioning and services provided in the marine system, but really is microbial biomass a used food source in benthic communities. I decided to kind of put together a three-part talk. I'm going to start out with a crabbing primer of biomarkers, which is a story of a new species of yeti crab. Then I'm going to go to the feature presentation. This I'm going to try to use kind of like the Pixar dancing lamp, so it should be entertaining. The feature presentation, which is the why, the title of the talk, etc. And then go to the prequel, which I think I learned from George Lucas, some of how you have to qualify many food web studies, because you know, if you tell a good story, you might as well release 3 more prequels and just ruin everything. And just So to start out, in 2006, I was on a cruise with 3 geologists, and they stumbled across this group of crabs at 1,000 meters, sitting, waving their arms at a methane seep habitat off the street. The geologists were not interested in them because they were not rocks. However, the subtitle said, "This is a brand new species, you need to take it to the biologist on board because he's going to say it's great." So they said, pull the subtitle, we'll do it quickly, and if you look closely, you'll see an arm come in from the side to grab a couple of them. There it is. And this new species was collected for the first time for science. Why it was especially exciting, and why the subtitle was super excited, was because this species, by some of my colleagues at Mbari as well as in France, was the first species of yeti crab, and it really captured the entire excitement of the world. There's yeti crab underwear, pajamas, more stuffed animals that I can talk about. Everybody wanted the yeti crab. So it was a new, at that point, super family within the Crustacea, and this appeared to be the second species within it, and it turned out molecularly that it was the second species in it. Why I was excited about it, being a trophic ecologist looking for different sorts of animals that use bacteria, is if you look at sea or tears, they're covered with these prebiotic bacteria. And so what are these bacteria for? There's been a lot of different species that have had bacteria growing on in hydrothermal vents and cold sea habitats, and they've never actually been able to be shown to be a food source. So I said, well, let's see if we can. So what do we need to say that bacteria growing on an animal is actually a food source? So it's getting energy coming up from the seafloor. So first off, we need to be able to say as chemosynthetic energy fueling its biomass. We can use stabilized top analysis for that. we need to be able to hopefully show that there's actually something within the bacteria that is traced into its tissue, showing it's actually consuming and integrating energy captured by that bacteria. And finally, it needs to have a mechanism to consume this bacteria. And if we're lucky, we'll be able to see it actually do it. And with all the main species that have been found and predicted to have epibionts growing on them, each one of these has not been the case. So the reason why I'm talking through this right now is because bacteria are small. And so if you look at an animal eating bacteria, how do you tease apart whether it's actually consuming bacteria using that food source or not? You can't just look at gut content because it doesn't tell you anything. You need to figure out a way to actually trace and identify a portion of that. And so we're going to do that through talking through the basics of stabilized glycan analysis, fatty acid biomarker analysis, and then just show some overview. The two biomarkers that I largely deal with are stable isotopes, which takes advantage of the ratio of carbon-12 and carbon-13 and nitrogen-15 and nitrogen-14. These are set in most cases by the producer and then largely directly incorporated into the tissue of whoever or whatever consumes those. The other technique takes advantage of membrane lipids, specifically fatty acids, and just like these, just like the stable isotopes, they're also directly incorporated from food sources, although with much more modification and alteration. However, this is two sources and two unknowns, whereas this can provide much greater resolution because there can be 60, 70 different fatty acids in food sources as well as grazers that eat those. The other thing I like about this slide is I made this figure for my master's students last time. So here's the more basic primer on stabilized toxicology. If you have two primary producers and you have a grazer, it will be able to have a isotopic composition that eats 50% blue triangle and 50% green star. That was the most difficult part of my talk, was to remember what color and shape those were. It eats 50% of those. It will be exactly in the middle, quantitatively saying it's eating 50% of each of these. Since these both have the same isotopic signature for del-15N, the ratio of nitrogen 15 to 14 compared to the standard, you're going to get a general increase just due to the metabolism of that species, which is freeze lighter nitrogen to experience magnetic. So this is the paradigm. And what we can see when we actually look at del-15N on the y-axis, del-13C on the x-axis, and we plot all of our plankton signatures from the Costa Rican margin, and we know that anything less than about 24 per mil is only explainable in the marine environment in that region of chemosynthetic activity. And we plot each one of these black dots, which is the tissue of a different individual of the kiwi specimens. You can see that there is a strong non-photosynthetic signal as the main source of food for the kiwi crabs or the yeti crabs in this habitat. Step one, check. Now, fatty acid lipid analysis takes advantage of these different membrane constituents. This is a 16 carbon fatty acid. It's got one double bond, 7 carbons from the end. And that's how you identify them. And it's not as much of a direct incorporation, but unique biomarkers can still be incorporated from different food sources. With a big caveat that all of us, all metazoans, all animals, can also have their own synthesis pathway, where they start with a 16-0 fatty acid. They can either add 2 carbons to it, going down the chain, or they can add a single double bond, 7 carbons from the end. And this is essentially the entire realm of what most animals are able to synthesize. And so if you combine unique fatty acids that will come from a bacteria, for example, and the synthesis pathway, you can come up with a good idea, although not quantitative, exactly what this individual is eating. On the y-axis here, I have percent of total fatty acids. On the x-axis, I have the dominant fatty acids within this particular species. Green is plankton, blue is the bacteria-covered seed tips, specifically this as a signal for the bacteria, and then black is the muscle tissue. And what we find is that compared to the phytoplankton, there's a far reduced amount of photosynthetic biomarkers, and most interestingly, the 16-2 is only found in a few main sources, including the bacteria-covered seed tip, as well as 2 other crustaceans that are found at hypertrophic habitats, Shinkai crosneri, as well as Rumicaris exophyllum. So that means that, first off, this was actually incorporated into the tissue, likely from the bacteria, but also those bacteria are a similar group that is found in hypertrophic species in mid-Atlantic ridge, as well as the Western Sea. Interestingly, one of the greatest challenges of identifying the ecology of these two species is they both have a high abundance of 16.2 within their epibiotic bacteria that neither of them actually incorporate that into their tissue, and that's why nobody knows whether or not these species actually incorporate the energy captured by the bacteria growing on them. So the other problem with Firmicutes exoculata and many of these other species, they have bacteria growing on it, but they have no way to actually grab that bacteria and put it in its mouth. And so we did some scanning electron microscopy, and here are the normal seed with their little tufts of bacteria, but they actually have specialized seed, which you can see in this photomicrograph, showing that they've been modified into cones, and so they actually have these modified hairs they use to be able to capture these bacteria and move them towards them. If you bring one up to the surface and you give it some time, you can see that on this particular arm, it's their maxilla, and it will start printing and cleaning and bringing those bacteria up to its second maxilla pegs or second mouth arms, and then essentially shoving those bacteria all the way down its belly. So essentially this was an example of just to introduce you to stabilized topic analysis and fatty acid analysis, but also there's this word crab that raises its arms to capture energy by the bacteria growing on it and fluid escaping from the deep sea. However, when we were down there, we also saw this, which we have no idea what it is, but it's essentially one especially furry crab with top. And the first time I gave this talk, a very senior and distinguished deep sea ecologist was in the audience, and he was the first person to raise his hand. And I called on him. I was quite honored that he would have a question for me. And his question was, "Have you thought about sex?" I had no idea how to answer that. Thankfully, he expanded on that and said that that could have been exactly when a spermatophore was passed to a female that had just molted, and so that might have been a reproductive dance and display, and then everything thought about in deep sea. It could also just be the craft playing King of the Hill. You know. On to the feature presentation. So the deep sea is incredibly seasonal. The reason why we go to the Antarctic is it's also cold. The bottom water temperatures, 1.8 C are common. Average depth of deep sea, 3.6, 4 degrees C, which is fairly cold by most standards, and incredibly seasonal. The Antarctic has incredible seasonality, and it's also cold at 1.7, minus 1.7, minus 1.8. So it's a good analog to understand the systems. Now it's nice to tell people that there's high seasonality in the Antarctic, but it's easier to show people. This is just after the sun's risen. This dive hole is about 120 feet away or so, and you can see there's absolutely nothing in the water column between the camera and that hole. Here is February, and this is actually after most of the phytoplankton has come in and left. And you can see from the sea ice community and the fuzziness throughout that the visibility has dropped due to a massive amount of photosynthetic production that's come in over a short period of time. In this particular region of the planet, that short period of time is about 1.5 to 2 months. And then it gone. We're talking about orders of magnitude, great differences in the amount of food source that's present. And yet, strangely, and thanks to some of John's earlier work, there's a unique polychaete habitat that has some of the highest density of animals on the planet. This is the Spiothenes tertianite bed. Each one of these tubes has a polychaete inside it, and the densities are sort of crazy at 150,000 plus individuals per meter squared macrophones retained on either 500 or in my case the 300 micron sieve. We also know from some of the work that John and Stacy have continued to do that this has been persistent for greater than 3 decades, and also if you do reduce the food supply of the habitat, the community shifts. So it's a food limit community that deals with incredibly episodic production and also lives in cold temperatures, making it kind of a perfect endpoint mimic for the deep sea, where we hope that we can actually see changes over a short time period beyond decades. So we wanted to see what role does bacteria play in this community. And first thing to do is ask, is there seasonality in the community, or is the same food source present throughout? Are bacteria used as a food source? And then finally, if we can demonstrate that, is it a necessary food source, or is it just a food source? There's one kind of classic, it's not quite a paradigm, but it would be a good paradigm because it's a neat idea to explain how these communities are able to deal with such intense seasonality, and that's called the food banks hypothesis, which is an acronym. And I was quite impressed that Stacy was able to say the INDEEP acronym, because I've been part of that since we started. I had no idea what it stands for. INDEEP's easy to remember, just like food banks is easy to remember, but 30 letters in a row, or 30 words in a row is hard to remember. Anyway, the idea here is that you get a seasonal deposition of phytoplankton onto the sediment surface through bioturbation of the fauna that's mixed down within the sediment, and importantly, and perplexingly somewhat, the idea is that the food stays there because bacteria to cold temperatures are inferior competitors and not able to break down this otherwise rich food source of labile or tasty food because it's buried in there, and so the fauna can essentially continue churning up that mud and consuming it throughout the year. To see that in more of a graphical form, you have December, February, May, August, November, 5 months of sun, 5 months of dark, and you have this incredible shift in food supply. And the idea is that because the bacteria are inferior competitors, you have a labile food source that's present throughout. So it's just carbon digestibility, graphical representation. And even though that labile food source decreases over time, it never shifts entirely over to a refractory food source, so there's always a good food source for the animals. What this means is that because the bacteria are inferior competitors, the macrofauna essentially means there's, compared to the macrofauna, there's very little or stable competition throughout the year between these different size classes. I don't know, I mean, that is a great idea, but I don't know how much I agree with it. And so the other alternative hypothesis is what I'm proposing is called microbial facilitation. And so we have the same change in overall production, but there's an important balance that as soon as there is a good food source on the sediment, both the bacteria and the worms, the anemones, and other things are both competing to try to use that food source as rapidly as possible. At some point, as the labile food source runs out, they're both essentially going full bore. They run out of that labile food source, and all of a sudden, you're left with refractory carbon, which the metazoans, the animals, can no longer consume. And instead, the animals then switch over and consume the bacteria. So essentially, it turns into best competitors, or competitors turning into facilitators in the seasonal cycle. The key here, though, is at the very end of winter when food stress should be the greatest. Because this is going to tell you whether the bacteria are an important food source and used food source or not. So we went out and we sampled in winter. We use these published from here on out. Winter, which is the very end of August, which was within 2 weeks or a week of the sun rising. We sampled this community, spring, and then also after the main photosynthetic input in summer, to look at essentially how does the natural topography of the food web shift over these different time periods. One of the ways we can do that is because I said that isotopes are a quantitative method. You can actually take the total area of the community. For example, each one of these points is an individual within the community there. And you can integrate under the total, essentially you just connect the outside dots to get the total area or niche stage occupied by the community. You then resample that and do some Bayesian statistics, and you can come up with an ellipse that represents the total area of the community in isotopic space. And if you do that, and it's called a CAC, if you do that, there's a non-significant but enticing shift going from winter with a broader niche breadth down to spring and summer, where it's nearly constant and appears lower. However, this is not significant. And I should say a couple of the key caveats there is in each one of those characterizations of the seasonal food web, you need to make sure you're sampling the exact same community, same species, and you do all of that. So these are completely comparable among season, and the communities persistent over decades, so there were no seasonal shifts in the community. So there might be something going on in winter just looking at this total integrated area niche. However, if you look at just the del 15N, which is also a measure of the trophic level, you can see a significant decrease in winter compared to spring and summer, which means throughout the winter, whether you're looking at the whole community, the dominant polychaete, the dominant anemone, or the winning polychaete, they all are feeding on a lower trophic level during winter compared to the rest of the year. So there's a seasonal shift in what the animals are doing. And that seasonal shift is essentially bacteria. Each one of these points is a fatty acid signature of a single individual of Scophens turturni, the main polychaete species. The only significant difference is going from winter to the rest of the year. There's no difference. This is not significant between spring and summer. And what's driving this difference between samples at the end of winter, beginning of spring and summer, is a decrease in the total bacterial biomarkers that are being incorporated into the tissue. So they're eating bacteria at the end of winter. Strangely, there does not seem to be a difference between spring and summer, even though there's a huge amount of productivity that comes in after that, between these two time points. And so what explains that? And that's actually benthic production. These are benthic diatoms growing on sediment surface. There's a micrograph of them, and they can essentially show up as soon as the sun rises, and there's very little light in the ice. To give you an idea of how much the total requirements, we did some in situ benthic respirometry using the old light-dark reaction, where we just taped up a sediment core, took it, and took another one, and amazingly worked. We didn't actually have benthic chambers on station because we didn't think this was going to be important. But just measuring this, we were able to get 25% of the data requirement shortly after the sun comes up is accounted for by benthic production. So it's not just phytoplankton, it's actually benthic diatoms, which immediately ameliorates the stress, the food stress provided by the 5 months of darkness that is the Antarctic winter. Okay, so microbes are a used food source, but are they a needed food source? And so to test this, we did a mesocosm experiment collecting, that would be anything, 92 separate sediment cores and providing 3 different food sources, 100% food, no food, and then 30% food. This 30% food was essentially the amount of food that we've known in the past to actually stress the community to the point where it deviates from its 30-year average. And so we know this is a stressful condition. And then we removed the microbial activity through gently pumping antibiotics through these cores. In many places, likely here, if you try to use antibiotics to knock out microbial activity, it doesn't work very well. And that reason is because there's so many antibiotics coming off of human activity, wastewater, and everything else. They're not working on. The Antarctic fauna is completely naive. So one of the viewers' main concerns about our proposal is that the antibiotics wouldn't be effective. We found every single antibiotic we tried to knock out microbial activity worked beautifully and essentially knocked all of microbial activity down. And so we very effectively reduced bacterial activity, and so we could see how with and without bacteria impacted the total topography of the food. This is what it looks like, all run in the dark. Yeah?
Is that a well-known fact
Which part?
that the penicillin, et cetera, antibiotics in coastal systems are in place and active right now?
Yep. It's in fact, you can even find them in the middle of the ocean, or you can find a lot of antibiotic resistance bacteria.
Thank you.
Here is our experimental design, each one with a separate flow-through system, and each one with a different food source, whether that 100% or 30% carried out in the dark. All on site, so there was no travel. We were essentially 10 minutes from the site, able to get the course in the lab and running smoothly. To tell you the rest of this demo, I need to have a little bit more information about isotopic analysis. So here's the essentially 0 to 1 shift in carbon that's incorporated into a grazer from a food source, for example, phytoplankton, and it increases by 3.2 is the paradigm. However, if something is starving, it can no longer be a food source, and so it starts eating itself, and you can get rapid spiraling off of the del-15N signature because it's essentially its new trophic level is consuming its own body. You can get very weird and high values of del-15N. It's been well shown from marine mammals and some other groups. So when we look at the del-15N of our different treatments, no antibiotics, no food, no antibiotics, 30% food, and no antibiotics, 100% food, we get some variation over the 6 weeks that we ran this experiment, nothing significant, and as you can see, the error bars are quite large. So a community that has normal microbial activity essentially doesn't care how much food it's getting at this particular time. In sharp contrast, if you reduce the microbial activity, all of a sudden you have antibiotics with 100% food, and this is very comparable and not significantly different from those with an intact microbial community. However, if you don't add food, you get these erroneous spiraling off del-15N values indicative of the communities actually starving because they don't have the bacteria to facilitate the long-term persistence. So what this shows is that because of these incredibly high values, this is a community that's in the process of failing compared to if you give it enough food, it has not yet failed because it hasn't broken down those food sources. Or if you just allow microbial activity to stay intact, it also is quite content to continue on. And at this point, the species have had, or the community has had, zero photosynthetic input for something like 7 months. So essentially, bacterial production, bacterial processes, or bacterial breaking down of refractory matter provide this necessary seasonal food supply that's critical for long-term persistence. So bacteria were used food source. There's a seasonal shift in the total topography of the food web, and inhibition of bacterial productivity led to starvation. You can imagine how bacteria are now integral in our understanding of synthetic food webs, because they're actually, even if they may be a small amount of production, they're needed to have that community persist. And that can impact the carbon balances on some of the largest habitats. So that's the good story. Now we have to get to the prequel. So one of the challenges of biomarkers is that these are paradigms built largely on meta-analyses of a very broad swath of the animal community and have not focused on microbial production. Microbes provide a variety of different building blocks and different energy sources than most animal communities. And so we wanted to see, do the caveats of these biomarker approaches actually ring true when you deal with microbial sources. I'm going to jump over to a different family of polychaete. These are the Doralated polychaetes. The reason why I decided to work with them for this particular part is because they are abundant in reducing habitats, and they're environments that have abundant bacterial and archaeal production, which is not always the case in other locations. And so as a family that we know eats bacteria, so let's see how they deal with bacteria. So, as I said, stable isotopes, quantitative measures of realized trophic linkages over extended periods of time, they have the ability to resolve microbial detrital food sources. There should be a question mark at the end of that, but they also lend themselves to use in habitats that do not allow easy study. Deep sea, reducing habitats, or any sort of habitat where you're trying to identify the incorporation of organisms that don't form microns in size. However, we really don't know if they're appropriate, and so the best thing to do is to use a family that's ubiquitous and a family that's been around for a long time and actually test whether these paradigms are true. The other nice thing about them is they form broods. This is a little brood of Ophryotropa veronica. It's a shallow water analog. When they're hatched up, they're this big, and when they grow up, they're this big. And I didn't put a scale bar, but this is a huge organism at about 2 millimeters in length. We essentially were able to split single broods into multiple different feeding trials. We fed them 2 sources of eukaryotic production, spinach and rice, which is actually what they've been raised on in the labs since about 1947. As well as 2 bacterial food sources, Bacillus subtilis, Bacterium profundum, and then 2 archaea, which has never really been shown to be a food source, but is a potential food source, Haloferus volcani and Halobacterium salmonarium. We essentially raised them from this side to this side and then looked at their biomarker signature and compared that to its food source. Now, I showed you starvation, I showed you kind of the average values, but this is, if you have a food source, you have 3.2 and maybe +1 in carbon, this is kind of the range that you're expecting based on large experiments such as this of non-microbial food sources and not panelists. You get this value for consumer eating this food source. There's ranges around that. It's not a single value, it's an average of many studies, and so if you look at the ranges, you can get this nice score spread. And when I looked at the isotopic signature, I was sort of surprised that these are the values I got. So each one of these points, there's a consumer that's been adjusted to be consuming this particular food source. The statistics for once were very easy. The prediction was completely different from the observed, and in fact, one of the most surprising things is there was actually a decrease in the del-15N signature from the food source. So that means any estimate of trophic position would be just completely wrong. I didn't believe this when I ran. I ran into 2 different isotopic ratio mass spectrometers. I ran into 3 different universities, and I ran into both Atlantic and Pacific water as the base water for these trials to make sure that there was not some sort of issue with the analysis. And none of that mattered. Instead of being just there's constant either 0 or 1 per mil shift in carbon between the food source and its consumer, it instead appeared to be a function of the isotopic composition of the consumer, as well as the C to N ratio of the consumer. And we were able to explain quite a bit of the variance as long as you were working with means. If you worked on individuals, you would spend much less of the variance, which means you need high sample size, and you also need to take into account the C to N ratio of the consumer. that wasn't that surprising. It was really this N15 shift where the black line predicted what you would hope it would be, and the green line, or the gray dotted line is what it was. Essentially, we had a very poor explanation of it, and we at no point had the 3 point we had a positive. At no point we had a positive shift, we always had a negative shift. So, to answer the question, are these appropriate? One thing that we didn't account for is we didn't want microbial growth. We wanted them only to eat the one food source we provided. So we provided antibiotics, essentially not having growth in these food sources. And one of the reviewers, normally we just all complain about reviewers. This reviewer had a brilliant idea, and he said, well, are they eating the antibiotics? And so I did a C-to-N balance and said, well, actually, the antibiotics are a relatively important food source, potentially. The other reviewers thought that was not the best idea, but I ran continued trials with, again, many of the food sources. Here we have in green with antibiotics, black, no antibiotics, and blue, the actual food source itself, del-15N, del-13C. And what you can see is that only for this particular, the spinach group, did there appear to be a potentially significant impact of the antibiotics. For the other food sources, there was no significant shift, although there was a lot of variance inherent in this method. Partially because these are incredibly small organisms, and I was also, each one of these experiments was as problematic as anybody can expect. We had the instrument break all the time, so we'd spend 8 months growing up the culture, send it off, and then RMS would break and lose all the data. So something that's not quite as replicated as I would like. However, what's really important here is that in no cases did the del-15N value actually increase from its food source. So that means when you're using microbial processes, maybe these are not as quantitative as we may hope. So it gets worse. The fatty acid signatures are very unique in certain microbial groups. Archaea, for example, don't have fatty acids, and one thing I can tell you is that archaea lipids are not incorporated into tracers. And the other thing to say is when you do a multi-dimensional scaling plot of individuals fed each one of these food sources, you can see there is no clear nor significant grouping of any of the different food sources. The fatty acid signatures using multi-dimensional analyses are essentially independent of their food source. Highly problematic for people in my line of work. And one of the things is this synthesis pathway that essentially provides the vast majority of the fatty acid signature to these forms, and synthesis pathways that are not known to exist in metazoa. Here we have prokaryotic lepronic that present deviation from mean. So we just took the complete average of all of the individual grouping for the fatty acid profile. And we essentially said, well, if you're fed this food source, how do you deviate from the mean? So you can see the nuances of different food sources. And then compare that to the food sources within the fatty acids themselves. And what you can see is if you squint your eyes really well, you can find a few things that line up. For example, this branch 15-0 fatty acid was slightly more expressed in the lebronica that ate that. However, it was present in all of the different food species. So it was getting that from some different source. 16-0 in certain food sources were highly more abundant when there was present in the food sources. But again, none of this could be predicted without any sort of a priori knowledge of the food source. And so it's highly problematic. Even more troubling was that the fatty acids within these worms included polyunsaturated fatty acids, which are not thought to be able to be made by any sort of animal life, except for a few very unique examples, including nematodes and now carpactiocopods, the only examples from that zone that can form them. That's why we're always told to eat DHA and EPA in our cereals, because we can't make them, so we have to get them from our food source. And yet these worms, even though we're not provided any of them, we're able to incorporate them. And while that's challenging to the overall idea of using these biomarkers to be able to tease apart the microbial food sources, it explains a lot of observations that we've had over the years using annelids and hydrothermal vents and methods. In many of these cases, we found polyunsaturated fatty acids at hundreds to thousands of meters water depth in organisms that we know are eating chemosynthetic energy. And for example, the red worm that I showed in that scheme has a polyunsaturated fatty acid signature whose isotopic signature is no way anything except for getting all of its energy from methane. So it explains a lot of what we've seen in the environment, but also presents challenges. So Del 13C was not 0 to 1 as we would get from meta-analyses, and what the literature, Del 15N varied between minus 2.7 and at the high, looking at not the means but the actual values, up to a total of 1.3, non-significant, and at no point was it 2.3 or 3.2, which is what's most common in the literature. The dominant fatty acid signatures in this particular worm were completely the result of de novo synthesis, and also this worm appears to possess enzymes which are not thought to occur from animals. Now, I would say that's troubling and that I should find it in my work. However, when you do these multidimensional analyses and you use the knowledge of the ecosystem, you can still look and figure out, especially using more advanced techniques, the relative input of different food sources to benefit food webs when we look at a whole. So if you look at the entire community, and I can give you other examples of that, you can still tease apart the relative input of different food sources. However, the ultimate goal is to come up with a quantitative mixing model based on fatty acids or isotopes. And I think from those data that I presented so far, you might be able to guess that those quantitative models are unlikely to represent at least this species, and it fits very well within our general knowledge of analytic ecology. And so, if you're looking at two species that are very slightly different fatty or stable isotopic signatures, when you're trying to partition that between something that has an isotopic signature of minus 22 or minus 24, and you say, well, let's call this, you really need to know the C to M ratio of your feeding trials to find out if you can even apply these quantitative techniques. Because right now, they really remain in the realm of qualitative practices. So that's what good science works. So, to finish up my discussion of the deep sea, the deep is vast and is at a area of research. And so this is something that is the next figure, 2 figures from now, I'll show you something that just came off my computer before I got on the plane. Very excited about it. And the real answer to this is that the deep sea vast, yes, but it actually means that people are so excited about collaborating that you can do incredible things just by collaboration, especially across national boundaries. Stacy mentioned INDEEP. I'm glad she remembered the acronym. But really, this is just an international consortium of deep sea scientists. We are not selected at all. So if you're interested in deep sea systems, look it up or get on the mailing list. It's a great group. But it also means if you send out an email and say, who has frozen sample from around the world sitting in a freezer and would like to work on a paper together, send it to me. People jump up, raise their hands, and ship things from around the world. And so this is the first run of this. Each one of these points, trying to look at ecosystem function deep sea, represents frozen sediment taken out of somebody's freezer and sent to me. And we wanted to see how variable the ecosystems are across those. Easiest way to do that right now is to use high-throughput sequencing and looking at the microbial community present in all these habitats. And these are all these habitats, meaning hydrothermal vents, methane seeps, oxygen minimum zone, shelf, slope, almost abyssal, Antarctic, and anything else, and canyon habitats, and seamount habitats. And we're able to look essentially from people's freezers how variable the communities are. This was done by Tuesday. She was an RE from Marshall University that spent her summer extracting 106 samples and coming up with a small data set of 9.7 million 16S reads from each one of these. Our average, or our mean, our workable read from each one of these communities, so each snapshot of each one of these communities is the 44,000 microbial sequence. So this is a huge data set to be able to look at the total functioning of systems. And this is not ready for prime time, but it's what I'm really excited about right now. Here we have each one of those family richness on the y-axis, depth on the x-axis. I have not yet had a chance to color code it by habitat, but you can see there's a general decrease with increasing water depth. And the more exciting thing is that each one of these water depths, there's incredible variability in total community structure. These are all from the same habitat. These are from all the different habitats. And so it's a snapshot into what we're going to be working on over the next few years as we really start teasing apart the immense data that we can get just from looking together in a way to approach this vast ecosystem and how it works. Lots of funding involved, lots of wonderful collaborators. And with that, I'd be happy to take any questions. And I'll just put this up because I really like it's a good way to lead the conversation. It's a great way to lead the ecosystem.
You spent some time talking about the seasonality in the anaerobic samples and about how production-based seasonality of production drives differences. I know it's open that up by talking about very deep sea hydrothermal vents, which presumably don't go through, I'm not as familiar with it, but the same seasonal cycle, or at least as strong. So what do you think is happening down in these deep areas that the cycle are more stable? Do they follow the same kind of seasonal pattern?
So, and that's actually one of the my third part. It's one of the most interesting things because for a long time they found polyunsaturated fatty acids in hydrothermal vent organisms. And they thought that they were taking advantage of the seasonal pulse of food raining down in fiber travel pulses. They didn't know that they could just make all this photosynthetic polyunsaturated fatty acids, but they were using them as a way to tie what was happening with photosynthesis down to the deep sea, and saying there was some sort of impact of seasonality on hydrothermal vents. But it doesn't appear to be the case. I think for hydrothermal vents, what's going on is there's so much productivity that releases the food stress. It instead becomes an area where temperature and competition for space becomes all important, and the seasonality no longer becomes important, except in certain areas where you have seasonal currents that can actually impact the amount of methane release from different margins. And so you do get seasonality, but it's become because there's a change in the water temperature, which leads to higher destabilization, methane release, and increased food sources of those, but it's not the actual rain for synthetic matter. Having said that, and thinking while I was talking, we did find this really cool community in New Zealand that we went and we never found microbial mat the entire time we looked. And that is the classic methane seep habitat as microbial mat. Instead, we found this wonderful community of polychaetes, so it made me a very happy polychaete taxonomist and ecologist. We went back right after there was a big fiber travel pulse, and all of a sudden, all these microbial mats came up. So what I think that was microbial mats were essentially under the surface with the fiber to travel pulse, provided a huge amount of carbon, and rather than actually helping the worms, what it did is it shifted that balance of power between the microbes and the rhizomes that led to increased sulfide production and actually moving out the sulfide-oxidizing bacteria onto the sediment surface. So the surface seasonality may actually be a hindrance to the animals because it shifts that delicate balance between oxygen and productivity by the microbes, allowing them to outcompete the worms.
Then do the metazoans eat the mat down?
Yep. They slowly fight it back. And that's, we don't know that for sure, but that's what it looks like from what few samples we have. And a few glimpses into that habitat.
So this question is going to be kind of general because I've sent so much data, I'm not sure which experiments put it in context of, but when you're using antibiotics in your experiments on these metazoans, what do you think the effect on the internal microbiome is? So the animals is not, you're looking at their consumption of external bacteria, and yet you're also potentially disrupting their internal microbiome, so their digestive process may not be normal, their physiological state may not be directly comparable to the control treatment, because that is could be a uncontrolled variable. Have you given any thought?
Absolutely. So the question is, are you altering the microbiome within the sediment tract of these organisms? Because that's not necessarily impacting, you might not only be impacting the free-living bacteria within the sediment, you might actually just be changing their digestive patterns. And yes, and the only way to deal with that is to say that we're dealing with microbial processes, including both free-living and those in the sediment, because right now there's no real way to deal with that without actually looking at changes in the microbiome over time and with these antibiotics. But then still, I have no doubt that they're actually going to modify that microbiome, so is that a significant impact or not? However, that doesn't necessarily change the question as to whether bacteria are important. It means they are important, but it very well could be those that are inside the gut rather than just those that are free living. And that's a very important question.
And to go to your observation that the worms have enzymes that are not expected in metazoans, and I think you pointed to Harpactoi copepods and nematodes, which are not terribly closely related to each other or to your annelids. So, but it could be that they share some aspect of the microbiome which is actually responsible for that enzymatic.
It could be that. It could also just be that people have not really looked, because using large organisms, using mammals, and using even more advanced invertebrates, they haven't found that. Partially, I mean, one of the great examples of the difference between things that have access to good food sources and not. Cow microbes cannot synthesize polyunsaturated fatty acids. Only Antarctic can. The reason why we know nematodes is because somebody actually looked. And so it might just be that we haven't looked. We've used these assumptions built on different groups and said they must apply down, but the more we look, the more we find.
Andrew, in that generic food web isotope fractionation plot where I think it's del N15 on the Y and del C13 on the X, the classic pattern to expect, because it's like a textbook explanation, is that the grazer or the predator becomes 3 or so parts per thousand richer in N15 and 1 part richer in nitrogen, and that's your expectation. So the food source leads to a consumer that's greater in the isotope ratio. And then you got the opposite when you force the organism to eat bacteria. Is that what I heard?
Yes.
Did I hear you say you don't know why?
We don't know the enzyme responsible. And the carbon I'm not surprised by. The nitrogen really I am, because although that's the paradigm for food web ecology, is that you get this 0 to +1 for carbon, plus 2.3 to 3.2, depending on which paper you use.
And isn't the logic behind that the lighter isotope is mobilized more readily and therefore it's acting as the denominator and both those ratios go up?
Yes.
What's the explanation for the opposite trend?
There is none. That's why it's really exciting. Because the general paradigm for isotope biogeochemistry is anytime an enzyme, if it selects at all from one of the isotopes, it's always going to select for mobilizing the lighter isotope first. So why are we getting heavier left?
Well, first of all, I find it hard to believe that with all of the nice work in isotope food web analyses, that nobody has investigated bacteria as a food source and what it does to the consumers that eat it. Is that really true?
No.
Andrew's the man.
Yeah, I was surprised too.
Have you been able to, is it technically feasible to look at assimilation efficiencies, actually pre-processing fecal pellets in your polykeep sort of model system? I'm just wondering if they're, just consuming and reprocessing their waste material, and if that could drive some of these changes.
Yeah, so that would explain it in some of the labs studies, is that they could be reprocessing. So when an organism consumes the nitrogen, that's a really great point. When an organism consumes a food source, it's going to retain in its body the lighter nitrogen, the heavier nitrogen, and expel the lighter nitrogen, leaving its fecal pellets heavier, because it's going to assimilate the nitrogen quicker from its food source, incorporate some of the heavier nitrogen and expel the lighter nitrogen, but coming out of that food source, it's going to be the heavier nitrogen that's left in its fecal pellet. That was a very confusing thing. Pretty much.
Yeah.
More than this in my head than talking. It's easier if you have a chalkboard. You guess what and so you essentially have fecal pellets that are heavier nitrogen. So if they're constantly remineralizing, consuming those fecal pellets, that could be a way that if they start breaking down further and further compounds that they weren't able to consume on the first round, they'll start having a food source that is lighter in nitrogen. Lighter in nitrogen. I actually need to put this on the board and work it out, but that is a way to do it. However, I will say that we always provided the food sources in ample quantities, and they have a see-through body wall. You could always see that they actually had the bacteria, archaea, or the eukaryotic sources in their stomach. Mm-hmm. It doesn't mean that it might not have been recycled, but that would suggest it's otherwise. But that's a really good idea. As soon as you get away from the simple +1 plus the 3.2, you can start getting into a kind of a whole lot of plausible or somewhat unplausible circumstances, one of which is probably true, but it's hard to tease those apart.
Yeah, especially in the trail community where there's ample crap around. And then I don't know the metabolism of the strains you're doing, but they're really mineralizing some of that waste.
We did kill and freeze dry the bacteria because we didn't
No,
want them
okay, so-
actively modifying the carbon pool and nitrogen pool within the solutions.
Okay, I think we'll say thank you to Andrew, since it's after 5.